Welcome back to my course aspects of biochemical engineering. Now, I was discussing about the kinetics of substrate utilization, product formation, biomass production using microorganism. So, we discussed the theory part as well as we discussed several problems, and coming to lectures also we will try to solve problems, because what I believe that if we can solve more problem, the conception of the on the microbial process will be more. How to analyze the chemostat, how to analyze the plug flow reactor, how to analyze the fed batch process. Now, so today this whatever lecture we have framed that we will try to go much details of this particular microbial fermentation process. The first problem that I want to discuss, if you look at this problem, this is a generalized form of the logistic equation is proposed by Konak, such as this is the equation. So we know, if we can remember the life cycle, so it is like this: time with respect to viable cells. Am I right? Viable cells. Now these viable cells we can express in terms of n. So it is like this: it is going, then stationary, then coming back. So here we consider we consider in infinity, or in then infinity means the cell mass constant, number of cell mass at the stationary phase. So this is the infinity is the stationary phase population, AB is the constant, this A and B are the constant, and N is the cell mass concentration at any time t. Now, what we shall have to do, show that the maximum growth rate occurred when then n by n infinity equal to A by A plus B, and that its value is given by this equation. So, if you put these values in this equation, that we can find out the maximum rate of that cell mass production, that should be equal to k into n infinity A plus B, A to the power A, B to the power B divided by A plus B to the power A plus B. So, this is very interesting problem. Now, now to solve this problem, what I want to tell, that we know that dN by dt, what is dN by dt? Rate of cell mass formation. This is with respect to time. Now, what will happen? This will keep on increasing with respect to time, then will decrease. Now, this we shall have to monitor. This is what is this point? This point is dN by dt. This should be maximum. So, this is also T max. So, at T max we will get the rate of cell mass formation is maximum. When it will occur? When d2n by dt2, this should be equal to 0, because it increases, then it attain the plateau. When it attain the plateau, then it will be maximum, and this is possible when d2 by d2n by dt2 equal to 0. Now, this is the solution how we try to work out with this problem. If you look at here, that this is the problem that has been given. Now, if I take n infinity A plus B in the right hand side, the equation will be becoming this. This the equation will get like this, and then then if we differentiate with respect to time, then by differentiation we will get this equation. So, and then after getting this equation, then we put these conditions. When dN by dt equal to max, the dt2 by dt2n by dt2, this is some mistake is there, you can correct it, this should be equal to 0. Okay. So, this I can explain you little bit to you that that this if you differentiate this, that that this is n this is constant, am I right? But this is n is the variables, and this is also variable. So, we differentiate with respect to these two terms, and when we differentiate with respect to time, this will come like this. Now, so as now if we put these conditions that d2 n by dt2 equal to 0, then we can write this equation in this form. Then the equation will be becoming simplified in this form, that a into 1 minus n by n infinity equal to b by n infinity. Now, when we go further, we find we will come across this equation, and this equation if we solve it, if it minus 1, if you take this side, then infinity, I can write that a n by n infinity equal to a plus b. That means, we can say at T max when d2 t by dn2 is equal to 0, and this is occurred when dn by dn dt is maximum. This will be, this is possible, then n by n infinity equal to a by a plus b. This we can easily find out. Now, this thing we can put it in the previous equation, that whatever we have, this equation we can put it this equation, that this equation we can put it, and if we put that this equation, we can come across, we will come across this kind of situation, and then if you simplify it, it will come like this: dn by dt maximum equal to k n alpha a plus b, a to the power a, b to the power b, a plus b equal to. So it is very simple problem. Only the thing is that we shall have to find out that under what condition the rate of cell mass growth will be maximum, and rate of cell mass growth will be maximum when d2 n by dt2 equal to 0. That is because I told you, I am repeating you again, the dn, the dt versus t when you plot. So, here is the maximum, and here at this particular point d2 by d2 n by dt2, this should be equal to 0. If you differentiate that, we can easily find it out. If you put this, you will come across this kind of equation. This is a very simple problem. I hope if you try, you can solve it by yourself. Now, another very interesting problem that I want to discuss here, that we know that we come across the Pirt equation. What is the Pirt equation? Pirt equation deals with the maintenance of the cell, all what you call endogenous metabolism of the cells. So, what is the, I told you what is the maintenance of the cells. Maintenance of the cells means in a solution, when you keep the cells, when you see the organism under the microscope, you find that organisms are moving from one place to other place, and this, for the movement of this organism, you require some energy, and for the some kind of protein formation or some kind of repairing of the ruptured cell, we require some kind of energy. So, that is used for the maintenance considered as the maintenance energy. So, what Pirt equation basically deals with, that they are saying the substrate mostly goes for the cell mass formation, but a portion that is used for the cell maintenance. Now, so this equation solely deals with that. Now, when a negative term is included in the cellular kinetics in order to model the endogenous metabolism or the maintenance energy, the resulting equation, assuming the excess substrate, can be written as that is. Now, this equation we have shown you, this is nothing but ds by dt, this is overall rate of substrate formation overall, this will be minus ds by dt, this is for growth, plus dA minus ds by dt, this is for maintenance of the cells. So, this equation we have shown you that this equation is nothing but this. Now, if you divide by mu x, then the equation will be can be written as what will be this? We can write 1 by y equal to we can write 1 by y dash plus k e by mu. Am I right? So, we can write this equation in this form. So, where y dash is the growth yield is the gram of cell produced per gram of substrate consumed for growth. Now, here we sometimes we call it true growth yield, true growth yield. What do you mean by true growth yield? Suppose we already have come across the yield coefficient. Yield coefficient means gram of cell produced per gram of substrate consumed. Now, if the substrate used only for the cell mass formation, then we call it true growth yield. Okay. Now, this is so this is the true growth, and this remain constant for the all organism, and K e is the gram of substrate consumed for the maintenance energy per gram of cell. Now, here rate of substrate consumption for the maintenance of the cells is what is always proportional to the concentration of the cell, because more cell, the more will be maintenance energy requirement, less cell, less will be the maintenance energy. So, what we can write that minus ds by dt, this is for maintenance, this should be proportional with the cell mass concentration. So, this should be equal to m into x. M, what is m? Maintenance coefficient. Now, here this has been substituted by K e. Now, K e is the gram of substrate consumed for maintenance energy per gram of cells. Now, y is the apparent yield, sometimes we call it overall yield coefficient, and gram of substrate consumed per gram of cell mass formation. So, what we shall have to do? We shall have to show that in a CSTR or chemostat, X and S are related by this. This we shall have to prove that this is related by this. And then what is the relation? What is the relation between K e, y dash and d in these circumstances? And does the figure of below the aspergillus aerogenes growing in the glycerol satisfy this model? So, they have you know that Aerobacter aerogenes, this is kind of organism that is that when you plot 1 by y versus 1 by d, d is nothing but the dilution rate, they get this kind of correlation, and we shall have to prove, we shall have to find out whether it satisfied this part equation, part models. So, this we shall have to prove. Let us see how we can solve that. Now, this is the basic equation is given that this is the part equation. We know this is the part equation. This is given in the problem, and we know mu equal to d in a chemostat under steady state condition, and the sterile feed. What is the steady state condition? Let me repeat it again, that steady state condition means when the concentration of the different material present in the reactor remain unaltered, remain constant, steady with respect to concentration. When it is changed, this is unsteady state. As for example, suppose you are putting the substrate in the reactor, and if the substrate concentration keep on changing, what do we call it? Unsteady state. Now, if the substrate concentration remain constant, this does not change with respect to time, then we call it steady state condition. Same thing applicable to the cell mass, same thing applicable to the product formation. So, that is the steady state. So, under steady state condition and sterile feed, what does it mean? When the media content does not have any kind of cell mass, x 0 equal to 0, then we call it sterile feed. Now, here this equation I can write like this, that we can divide by x, then we have this. If you divide by x, then we will be having the equation what mu by y equal to mu by y dash plus k e. Am I right? And then d equal to mu. So, if you replace mu by d, so we will get this equation. and finally we will come across this equation. So, what the problem that we have this kind of equation, then what we know y is the overall yield coefficient or average yield coefficient. This is equal to x minus x 0 by s 0 minus s. Now, then what we can write, d by d y dash by k equal to this yield coefficient. So, if we put sterile feed x 0 equal to 0, then x can be written as this is multiplied by s 0. So, this is what a portion of the problem is looking at that given that you have to prove that x equal to y dash d s 0 minus s divided by d plus k plus y dash. So, this can be easily proved. Now, question comes, what is the relationship between d, ke and k dash. So, what we can write that that what we have here what we have find out y equal to, y is the yield coefficient, am I right? This is equal to d by d y dash plus k. Now, y yield coefficient always less than 1. So, we can always write this is less than 1, am I right? This is exactly what is written there. Then if we do the reverse, then if we bring it at the top, then what will happen? This will be greater than 1. And then we can have this relationship as have this relationship that we have. This is that 1 by e. Now, this we can do this further simplified, that what we can write, this is d plus ke into y dash by y dash into d, am I right? This is like this. I can write like this. Now, if I multiply both side y dash by d, then it can be written as d plus ke y dash, it should be greater than y dash d, am I right? Then if you if you divide by y dash, then what kind of correlation you will get? Y dash, if you do that, then d, then the same kind of relationship there. So, this kind of correlationship that hold goods. We can write that this kind of correlationship between the d, k and y dash, we can have this kind of correlation, or you can divide by y dash, then you can bring this one term this side and make the things a little bit simpler. So, this is how we can find out the correlation between y dash, ke and d. And last part of the problem that we have, that in case of Aerobacter aerogenes, they found the correlation between y 1 by y by 1 by d, because this is the equation we have, because if you look at we had this equation, am I right? This is the equation we had. Now, if you divide by mu x, then what will get? Y 1 by y equal to 1 by y dash plus k into mu. Okay, am I right? So, this is exactly what we wanted to do it here. We have only this. We know under steady state condition and sterile feed, the mu equal to d. So, if you put mu equal to d, so we can write here d. Okay, this is only the difference. Now, here what are the variables? Y is the variables of overall yield coefficient and d is the variables, and ke is the maintenance coefficient and the y dash is the true growth yield, that are the constant. Am I right? Now, if we plot 1 by y versus 1 by d, then they found this is kind of correlation they obtained in the Aerobacter. But as you know that if you look at this equation, what is this equation? The equation equal to y equal to c plus mx, and this is basically a straight line equation. So, it should be a straight line. Instead of straight line, this is a kind of hyperbolic type of equation, the kind of relationship that we have. So, what I want to comment here, that this relationship does not satisfy this model, because this what is the curve that is showing here, that that is not satisfy this model. That should be the answer. Okay. Now, let us go to the that another problem that is also very interesting, that that a microbial, because in case of microbial fermentation process, we have come across different type of inhibition. We have come across the substrate inhibition, we have come across the product inhibition. And I told you, you can remember, in case of substrate inhibition, usually we prefer the fed batch reactor, but in case of product inhibition, we preferred the plug flow reactor, because why? Because we have seen that that when you plot 1 minus R A versus C A, this kind of plot we have. That means, when substrate is degree, this is C A 0, am I right? And this is your C A. This might be your C A. So, when its substrate concentration decreases, that 1 by R A increases, that means R A decreases. Now, as the substrate concentration decreases, why the rate of reaction decrease? Because mainly due to the product inhibition. And we have seen that in case of this, if you have this kind of correlation, we find the area under this curve. This, what is the area under this curve? Is tau plug flow reactor, am I right? So, this is much less as compared to this is this, what is this? This is the area under this, this is the rectangle, what you call tau CSTR. So, tau CSTR will much higher as compared to tau plug flow reactor. So, we will prefer tau plug flow reactor, am I right? So, in case of product inhibition, we usually recommend plug flow reactor. Now, this particular problem deals with some kind of product inhibition, and we shall have to develop some kind of equation out of that. Let us see how we can do that. In a microbial species is inhibited by the volatile products such as the ethanol. We know that a high ethanol concentration, that growth rate of the organism that will be inhibited. Now, growth rate can be increased by the removal of the inhibiting product via continuous evacuation of the vapor space above the fermenter. So, what we can do, that that if we applied vacuum, we know the boiling point of ethanol is less as compared to that of water, am I right? So, if we applied vacuum, that even at the normal temperature, that alcohol will be vaporizes out. Now, if alcohol is vaporizes out, then that product inhibition due to certain concentration of ethanol can be overcome. Okay, that is exactly they have mentioned here. Now, for a batch fermentation process, following equation is there, that mu equal to mu max S by Ks plus S, Kp plus Kp plus B, and show that the time course of the substrate level, the overall is this S equal to 0.3 P plus cell mass. And if S is like this kind of correlation, if it holds good, then that then the equation will be this equation. That equation will be this equation. This is the ds by dt equal to x 0 plus y x by x, x 0 minus x, y by x by s, mu x, and s by ks, and this is the equation, and ki is the kind of inhibition constant that we shall have to prove. This is the equation, this is the problem that we have. Now, let us see how we can solve it. Now, this is the stoichiometry is given. The stoichiometry is like this: S equal to substrate equal to the 0.3 P plus x. The x is the cell mass, and this is the equation that is given. And we know ds by dt, how we can write ds by dx and dx by dt. Am I right? And dx by ds is equal to yx by s. That we can always, this is called yield coefficient. And dx by dt we can write mu into x. is the rate of growth of the cells. Now, here that so ds by dt, what we can write, that 1 by this and this thing I can substitute here, that in place of mu I can substitute into x. This we can get this, the two equation we can substitute, we can get this equation. Now, y x by s is x minus x 0 by y 0 minus s. Am I right? So, x equal to what? X equal to y x by s, s 0 minus x. Now, if we put this in the previous equation, then this equation, if we put it, then what we will get? We will come across this equation, ds by dt equal to 1 by y x by s mu x, s by k s plus s k p plus, and this x will be substituted by this. I can write like this. Now, this will cancel, and this and y p by s, how we can write? This is the gram of product formed per gram of substrate consumed. P minus P 0 by S 0 minus S. So, P equal to, I can write y P by S is 0 minus S into plus P 0. Am I right? And P equal to, the stoichiometry is given. If P equal to 0, then we can write P equal to 0, we can write, then P equal to this the 0.3 S 0 minus S 0 minus S, that we can write. The equation, if you look at the equation before, that what we have done before, that that this equation. So, we have this S equal to 0.3 P into x. Now, this equation we can put it here. So, this equation we can write in this form. Then we can put this value in this equation. Finally, we will come across this kind of equation. So, we will come across this kind of equation. I hope this is how we can solve this problem. Now, another very interesting problem is that we have come across the generation time. Am I right? And we come across the doubling time of the cell. Now, when we talk about the doubling time, what is doubling time? The time required to double the cell population. What is generation time? The time required for the one cell division. Now, here if you look at this problem, that a new microorganism has been discovered, which as its cell division, cell division means this is the generation time, it gives the 3 dotted cell, and then from the growth data below, calculate the mean time between the successive cell division. That means, we shall have to find out the generation time, Tg we shall have to find out. Now, in the doubling time, we have seen, what is the equation we have seen? D ln x by x 0 equal to mu into t. Am I right? Now, in case of generation, that doubling time, this will be 2 x 0. Then the equation will be ln 2 by mu, this will be equal to T d. Am I right? Now, here in case of generation time, what will this is called, this is one cell produces 3 cells in one division. So, equation will be little bit different, ln 3 by mu. Am I right? Now, question comes, how you can determine the value of mu. Now, we have here in this problem, what is at the different time, this is the batch system, we can easily estimate the cell mass concentration. This is the cell mass concentration we have. Now, from that we can now what is the equation that we have, that ln x by x 0 equal to mu into d. Am I right? Now, so by here if we assume this is mu is constant, because in the log phase, so we can write that x by x 0, we can write this value, and this value you can plot in the ordinate and t in the abscissa. Then we will get a straight line, and slope, obviously slope will give you the value of mu. Slope will, this is exactly what we can find out the value of mu. So, you can easily find out the value of mu. And then as I mentioned that in per generation time, So, what is that equation? This is like this: ln Tg, I told you Tg equal to ln 3 by mu. So, we have already estimated the value of mu. We put this value of mu and 3 ln 3 is there, then we can find out the generation time. So, this is a very simple way we can easily determine the generation time of the cells. So, in conclusion, what I want to tell that in this particular lecture we try to understand how to find out the time at which the maximum rate of growth of the cells take place, and we have given you some kind of logistic equation, that there is some kind of equation proposed by Konak, and from that equation we find out that Tmax value and how the maximum rate of cell mass formation can be expressed, that we try to discuss. Then we discuss some kind of product inhibition problem, and we try to develop some kind of equation, and lastly we try to discuss that kind of generation time of the cells. Thank you very much.